Good afternoon. And today we'll talk about the different types of computer-aided drug design. Okay. So we have talked a bit about the two different types of drug design, and today we'll talk about in details. Okay. So there are two major types of drug design. The first one is referred to as ligand-based drug design, or LBDD, and the second one is structure-based drug design, that is SBDD. Okay. So what are the differences between structure-based drug design and ligand-based drug design? So we'll just look at those differences very quickly in this slide. So structure-based drug design is also called as direct drug design. Okay, structure-based drug design is also referred to as direct design, whereas ligand-based drug design is also referred to as indirect drug design. Okay, indirect drug design. So the difference is that in structure-based drug design, as the name suggests, we directly are involved in the design of ligands for binding to the receptor structure that is known. Whereas in ligand-based drug design, we indirectly design the molecules or ligands which are going to bind to the specified specified receptor, which does not have its structure that is known. Okay, so the structure of the receptor is unknown over here. Okay, in ligand-based drug design. So, structure-based drug design, it relies on the knowledge of the three-dimensional structure of the biological target obtained through methods such as X-ray crystallography or NMR spectroscopy. So, receptor structure-based drug design, as I was just about to talk to you, as structure-based drug design is also called as receptor structure-based drug design. So, in this, we essentially know the structure of the receptor. Okay. Whereas in ligand-based drug design, we do not know the structure of the receptor. Okay. Hence, this is also called as receptor structure-based drug design. So it relies on the knowledge of three-dimensional structures of the biological targets, which are obtained through methods such as X-ray crystallography and NMR spectroscopy. So X-ray crystallography basically is the most widely used method for finding out the structures of the three-dimensional structures of the proteinaceous targets. Okay, but this particular method is often combined with NMR solution spectroscopy. Okay, so NMR spectroscopy is basically a method which exists in two forms. One is solid state and the other one is solution state NMR. Of which solution state NMR is used to predict, or rather to find out the structure, the 3D structures of the proteins. Now there is a difference between these two methods. In X-ray crystallography, not all the proteins may be amenable to formation of, they cannot be crystallized. They are not amenable to formation of crystals, which can be examined by X-ray crystallography. Okay, so we are calling this method as crystallography, right? So those structures, the proteins have to be condensed into crystals, and then X-rays are passed through those crystal structures. And then the X-rays, the images that come out are deciphered. They are calculated mathematically. That is how the protein structure is checked in X-ray crystallography. And X-ray crystallography is a very precise method for finding out the three-dimensional structures. Okay, it is that precise that even we can understand where is the binding site, how it is, how the intrinsic ligands are binding to the receptor. Okay, to the proteinaceous receptor. So X-ray crystallography is very widely used. In cases where the proteins cannot be crystallized, then there is a limitation for some of the proteins that they cannot be crystallized. And in such cases, NMR spectroscopy is used. And NMR spectroscopy has a drawback that it cannot find the binding site of the molecule. Okay, it cannot find out the binding site of the molecule. So that is why X-ray crystallography is the first choice for determining the three-dimensional structures of the proteins or of the receptors. Okay, and this is how the crystal structures or the protein structures are found out. And then those protein structures are used for structure-based drug design. Okay. Whereas ligand-based drug design, it relies on the knowledge of the other molecules that bind to the biological target of interest. Okay. So here we should know what are the structures of the ligands which are binding to the biological target of interest. And then these other molecules may be used to derive a pharmacophore model that defines the minimum necessary structural characteristics of a molecule that it must possess in order to bind to the target. Okay, so this is basically a inverse method, inverse method of drug design or indirect method of drug design. How? Let us just try and understand. Okay, so for instance, I'm saying that this is the structure of a molecule. Again, let us go back to a naphthalene kind of structure. Okay, so this is the basic structure of the molecule which is going to bind to the a particular receptor. Say suppose the receptor structure is not known. Okay, now but we know the ligands which are binding. So this is an naphthalene ring system. Okay, now say suppose an electron withdrawing group at first position is essential for activity. Electron donating group at 1, 2, 3, 4, 5, 6, 6 position is important for activity. Okay, now this is what we find out from the SAR studies, for example. And then, and then maybe a sterically bulky group at fourth position is important for activity. Okay, so what does that mean? Now, electron donating group is important for activity, electron withdrawing group is important for activity, and then a steric bulky group is important for activity. Maybe a charged moiety, a charged functional group is important for activity at this position, seventh position. So what can we infer from the SAR of these molecules? That the receptor structure is such that it can accommodate a electron withdrawing group over here, electron donating group over here, a bulky group over here, right? A bulky group, a functional group that is bulky over here, and then some other group over here, right? So which can be charged. For instance, if we are having COO minus over here, so that means that part of the receptor must be carrying some positive charge, right? So that it can interact with the COO minus that is present on this part of the molecule. So what have we done? By knowing the SAR of the molecules, we have indirectly tried to find out what is the structure of the receptor. So it has a binding pocket over here for electron donating and withdrawing groups. Okay. And then it has a large pocket over here which can accommodate bulky functional groups and a positively charged pocket over here which can bind to negatively charged functional groups of the molecule. Okay. So what have we done? Indirectly, we have found out we have tried to predict the structure of the receptor. Got this point? So indirectly, what we have done is we have tried to find out the structure of the receptor. Now, next, when I have to design a newer molecule, I'll design it in such a way that it has an electron withdrawing group, electron donating group, a very bulky group over here, okay, and a negatively charged group over here. So I can change the code structure now. I can change this from naphthalene to some other moiety. But what will I do? I'll ensure that these particular functional groups are present at these positions, right? So that is the reason why we call this ligand-based drug design as indirect drug design. I hope this is clear to all of you. Okay. So in receptor-based, structure-based drug design, we know the structure of the receptor. Okay. And then we try to design a molecule on the basis of that particular receptor structure. Whereas in ligand-based drug design, we do not know the structure of the receptor. We just know the structure of the ligands which are binding to the receptor. And indirectly, what we are doing, we are trying to find out or predict what would be the possible structure of the receptor. And hence, on the basis of that, we'll try to find out or we'll try to devise newer molecules, which may or may not be structurally diverse, but they will definitely bind to the receptor. So that is called as ligand-based drug design or indirect drug design. Okay. Then for structure-based drug design, if an experimental structure of the target is not known or not available, it may be possible to create a homology model of the target based on the experimental structure of a related protein. So this is called as homology modeling. Okay, this is called as homology modeling. So structure-based drug design may, if say suppose we do not have the X-ray crystal structure or the animal structure of the given protein, then what we can do is we can create a homology model of the given protein on the basis of closely related proteins. Okay, on the basis of some other closely structurally related proteins. Okay, so that is homology modeling. And then using the structure of the biological target, candidate drugs that are predicted to bind with high affinity and selectivity to the target may be designed using interactive graphics and intuition of the medicinal chemist, intuition and the experience of the medicinal chemist, the prediction ability of the medicinal chemist. Alternatively, various automated computational procedures may be used to suggest new drug candidates. Okay. So here what we are doing, we are using the structure of the biological target, which is obtained either via X-ray crystallography or animal solution state, you know, animal solution state structures, and or maybe homology model structures. Okay, and then we'll try to design molecules which will bind with high affinity and high selectivity to this particular structure of the receptor. Okay, so that is what is structure-based drug design. Let us go back to ligand-based drug design. Now, ligand-based drug design, what we have said is the receptor structure is not known, although the ligands are known. Okay. Now, if we have, highly active ligands, moderately active ligands, and inactive ligands, we can use all of them to find out what is the probable structure of the receptor to which the ligands are binding, and hence we can devise newer molecules which will be, good NCEs or probable drugs which will be acting against that particular target, right? So, a model of the biological target may be built based on the knowledge of what binds to it, and this model in turn may be used to design new molecular entities that interact with the target. So here in this example, what did we do? We took naphthalene based, naphthalene kind of molecules structured, I mean those molecules which have naphthalene in their structure, which are closely related to each other, and then we tried to find out in which pocket electron donating, electron withdrawing, bulky and charged groups are binding. Okay, and on the basis of this, what we have done is we have essentially found out a possible receptor structure. Okay, now this is a possible receptor structure. This is called as hypothesis. Okay, this is called as hypothesis of the biological target to which the ligands are binding. Okay, so this is what you are mentioning here, a model. So this is a hypothetical model. Okay, a model of the biological target may be built based on the knowledge of what binds to it. यानी कि जो naphthalene based molecules थे जो इस target को bind कर रहे थे उसके basis पे we have found out this model, and this model in turn may be used to design new molecular entities that interact with the target. Okay, so now we have this model which we know is binding to these naphthalene kind of molecules. So we can have some other kind of molecules. Say suppose simple phenyl ring based molecule. Okay, simple phenyl ring based molecule. So what is the necessity now? Necessity now that it should have a sterically bulky group over here. It should have a possibly a negatively charged group over here. It should have an electron withdrawing and electron donating group over here, right? So this is how we have just created a new molecule. We have designed a new molecule out of the knowledge of this particular model that was devised for naphthalene kind of molecules, naphthalene based derivative, right? So this is what is ligand-based drug design or indirect drug design. Okay. Alternatively, there are other methods also which are used in drug design. Okay. So for instance, QSAR, that is quantitative structure activity relationship. Okay. Now we all have studied about SAR in our earlier standards in our B farm maybe. Okay. So SAR is what structure activity relationship. But here we are talking about QSAR. Q indicates quantitative structure activity relationship. Okay, quantitative structure activity relationship. And in quantitative SAR, that is QSAR, what do? We develop a correlation between the calculated properties of the molecules and their experimentally designed biological activities. Okay, So these QSR relationships in turn may be used to predict the activity of new analogs. All right. So again, let us try to understand what is QSR. Now we know, that particular molecule. Let us take the example of naphthalene again. Okay. So if now we are talking about naphthalene kind of molecules again. So if naphthalene has an electron withdrawing group over here, okay, say suppose we'll try to find out some of its physicochemical properties, like for example, log D or log P. Okay, for example, log P. So if we have an electron donating group over here, it's log P के ऊपर उसका क्या effect होगा? Okay, what will be the effect of electron donating group substitution at first position of naphthalene on the log P and also the activity? Okay, we are also have to check out what is the effect on activity. Okay, I'll just write active over here. Okay, activity. So ये जो substitution इसमें introduce किया naphthalene में, what effect it has on the log P and hence what effect it has on the activity. Okay, now say suppose we'll talk about some other substituent, a naphthalene with a bulky substituent over here. Okay, maybe third butyl or isobutyl something. So bulky substitution at this position, what will that have? What effect will that have on the log P and hence the activity? Okay, then say suppose this position पे electron withdrawing group or donating group. This was withdrawing, right? So this is donating. So electron donating group at this position. What is the effect on log P and activity? Then negatively charged group at this position. What will be its effect on the log P and the activity? Okay. So what we are doing in SAR, what did we do? हम लोग ने सिर्फ ये बात किया था कि electron donating or withdrawing substitution at this position, negatively charged group at this position, sterically bulky group at this position. What effect will it have on the activity? So in SAR, we were talking essentially only about the effect of substituents on activity. But in QSAR, what we'll do, we'll first check the effect of substituents on the log P, and then we'll try to correlate it to the effect on activity. Okay, so understand this once again. Okay, what effect a substitution will have on the physicochemical property, and in turn what effect will that substituent have on the activity. So here what we are trying to do, we are trying to correlate a physicochemical property with the activity, and hence this is called as quantitative structure activity relationship. Okay, तो जब SAR कर रहे थे तो SAR में we used to just say that substitution ये इलेक्ट्रॉन विड्रॉइंग के बदले इलेक्ट्रॉन विड्रॉइंग दूसरा कोई सब्स्टिट्यूएंट डाल दिया तो इट इज गुड फॉर एक्टिविटी. बट नाउ व्हाट वी आर सेइंग, अगर हमने कोई इलेक्ट्रॉन विड्रॉइंग सब्स्टिट्यूशन डाल दिया तो इट विल हैव एन इफेक्ट ऑन द log P by this much amount, by a quantitative amount. And that quantitative amount, how that quantitative amount is going to have an effect on the activity. So what are we doing? We are trying to quantitate. We are trying to quantitate the effect of substitutions on the activity. And this is how QSAR was developed. Okay, this is how QSAR was developed. So using QSAR, we can quantitatively estimate, okay, as to what would be the predicted biological activity of the newly designed molecule. Okay, so after doing all this, we get an hypothesis, right? And then we'll try to generate newer molecules. So say suppose someone from you says कि नहीं यहाँ पे third butyl डाल देते हैं. Okay, some other person will say यहाँ पे will have another phenyl group. Some other person will say, why phenyl group? Let us have a bulky naphthalene group over here. So this will be binaphthyl type of compounds, right? So this way is will produce newer design molecules, and using this hypothesis, we'll be able to predict what will be the activity of those newly designed molecules. Okay, so that is what we have written over here. So these QSAR relationships in turn may be used to predict the activity of the newly designed analog. All right. So this is how ligand-based drug design or indirect drug design can be used for computer-aided drug design. So these are the two major types of drug design, okay, or the classification of drug design: structure-based drug design and ligand-based drug design. Structure-based drug design में we have the example of homology-based drug design, and we have the example of docking. Okay, you all must have heard of docking, or maybe few of you must be using it. Docking, this particular kind of software, they're called as docking. Or then we have something called as molecular dynamic simulation, MD, molecular dynamic simulation. So these are examples of structure-based drug design. And ligand-based drug design, mein, we have talked about pharmacophore model. Okay, pharmacophore-based drug design. So that is called as P, B, D, D. Okay, pharmacophore-based drug design. And then we have something called as the QSAR. So pharmacophore-based drug design and QSAR are two examples of ligand-based drug design. All right. All right. So then let's move ahead now. Whatever I had mentioned in this slide, I have repeated it here. Okay. So basically, ligand-based drug design is also called as indirect drug. So indirect drug design, and it relies on the knowledge of the other molecules that bind to the biological target of interest. So we have pharmacophore-based drug design and we have QSAR, that is quantitative structure activity based drug design. The structure-based drug design may structure-based drug design is also called as direct drug design, and it relies on the knowledge of the three-dimensional structure of the biological target obtained through methods such as X-ray crystallography or NMR spectroscopy. So this is again the same that is mentioned in the previous table. Now let me go ahead and talk about the different current methods that are available for SBDD. And these can be roughly divided into three main categories: the first method, second method, and third method. The first method is also called as virtual screening. Okay. First method is also called as virtual screening. So what do we do in virtual screening? We identify new ligands for a given receptor by searching large databases of 3D structures of small molecules to find those fitting the binding pocket of the receptor using fast approximate docking programs. Okay, docking programs, and this is known as virtual screening. In short, अगर बताना है, तो I'll just draw a structure of a receptor over here. Okay, now this is the structure of a receptor, and we know that this is the binding. This is the binding pocket. Okay, so what we'll do, we'll take databases of 3D structures of small molecules. Okay, now these databases can be, several billion molecules contained in that particular database. Okay, so so those billions of molecules will take, that their three-dimensional structures will take, which are energy minimized, and then we'll try to dock all of those molecules one by one into this particular binding pocket. Okay, so this is called as virtual screening. So what will we obtain after this particular exercise? We'll obtain a ranking of those billions of molecules. So few molecules will be, not binding at all. Few molecules will be binding okay, and few molecules will be binding very nicely and tightly with this particular binding pocket of the receptor structure. Okay, so those molecules which are binding very nicely and tightly with the receptor structure should be taken up for further optimization. That is called as hit optimization, right? Hit optimization, hit to lead optimization, and lead optimization. These are the three stages of drug design. So we had talked about this in our previous slide, in one of our previous lectures, wherein we said that computer-aided drug design can be used at any stage of drug discovery, right? Let that be hit discovery or hit to lead optimization or lead optimization, right? So this virtual screening or docking programs, these are usually used in the hit identification stage, but not necessary that they can be used only in the hit identification stage. If the docking protocols they are validated, then they can be used further in the lead optimization stage as well. Okay, and then the second category is de novo design of new ligands. Okay, de novo design of new ligands. Yeah, I was talking about the de novo design of new ligands. Okay, so this is the second category. In this, what we do, we know the structure of the receptor, right? We know the structure of the receptor. So what we'll do, we'll try to design new ligands which will fit into all parts of the receptor. For example, if we have a if we have a this a methyl group over here, or maybe a ethyl group over here, okay, which will bind to this pocket of the receptor. We can have a phenyl group over here which will bind to this part of the receptor. We can have a this, since this is a big pocket, we can have something like a fused aryl. Okay, so a phenanthrene, anthracene, or simple apna naphthalene type of molecule. Okay, so what have we done here? We have just found out that a ethyl fragment over here, a phenyl fragment over here, and a naphthyl or a anthracene or a phenanthrene type of a fragment over here will bind into the active pocket of the or active site of the receptor very nicely. Okay, so in turn, what have we done? We have done ligand design, right? And to be precise, this is de novo ligand design. De novo यानी starting from zero, right? So initially we did not know anything कि कौन सा ligand will bind, but now then we thought कि यहाँ पे ethyl group होना चाहिए, यहाँ पे phenyl group होना चाहिए, यहाँ पे naphthyl group होना चाहिए. Okay, and what we are having now is a newly designed ligand which has three different types of fragments which have been added to make up the ligand. Okay, so this is called as de novo design of new ligands, or in other words, it is also called as FBDD, fragment based drug design. Okay, it is also called as fragment based drug design. So ethyl fragment, phenyl fragment, naphthyl या anthracene या phenanthrene type of fragment हम लोगों ने एक साथ fuse कर लिया, and what have we done? We have produced a new ligand altogether, that is called as fragment based drug design. Okay, and in this method, ligand molecules are built up within the constraints of the binding pocket by assembling small pieces in stepwise manner. These pieces can be individual atoms or molecular fragments. The key advantage of such method is that novel structures not contained in any database can be predicted, can be suggested, can be designed. Okay, so the first method में हमने क्या किया था? Database of molecules use किया था. So those structures were already known. But here we are designing novel structures. So these structures are not known. Okay, so that means there will be a scope for IPR, intellectual property rights, in the form of patenting, right? For those pharma companies or those academicians who are going to use FBDD, fragment based drug design. Okay, so fragment based drug design, docking, these are all methods of structure based drug design. Okay, so a third method is optimization of known ligands by evaluating proposed analogs within the binding cavity. Okay, so now when we know that a certain ligand is binding to a binding pocket, okay, so what we can do, we can optimize the different parts of that ligand. So say suppose this ligand was having a ethyl group over here, but now we see that this is a quite a bulky space in the active site. So what we can do, we can add a third butyl over here, or maybe a n butyl over here, okay, or some other kind of functional group, maybe a phenyl group over here, that can accommodate the that can be accommodated into this bulky pocket, right? So that is how this third method is analogging. Okay, analogging. So we evaluate proposed analogs within the binding cavity. So once we design these ligands, we can again subject them to docking, and we can find out as to how those molecules are docking into the active site of the receptor with better predicted binding affinity or with lowered binding affinity. Okay, so that is analogging. Analogging can also lead to IPR. Okay, it can also lead to patents. Talking से patents बनना थोड़ा सा difficult है, but it is not that it cannot happen. But yes, we can have analogging and docking and fragment based drug design. Okay, the software used for molecular design and modeling we'll see in the coming lecture. All right. So all of you, just a moment.